Oh, right.
Yes, he did call me yesterday.
All right. That sounds like my friend Barney's having a job interview. Or something similar. Oh, right. Sorry about that. This is funny. I sometimes feel like I'm in some kind of, I don't know, like game that I don't realize I'm participating in. And every so often, like the games master, like gives me another challenge or something. I feel like I'm a puppet because my strings are being pulled. Anyway, unfortunately, Barney's dogs are not around because they're really lovely, but we'll just have to skip that. So again, sorry for the crazy start. I had to stay online with the tech because actually my wife's job is, believe it or not, even more critical of internet than mine is. I just wandered down the road to check out my friend's internet. Hopefully he'll make me a cup of coffee too. That'd be really nice. But my wife is kind of tethered at home, so she's kind of stressing out a bit right now. Oh, right. So I haven't actually done anything about checking where we were at. We were off starting in transposons, right? Where did we leave up? Actually, I could check probably a little quicker than I can get out of it. Move. Give me a second. That one. Goes. This is how I figure out where I'm at because I can never remember between different sections. At least he's got nice fast internet. That's a plus. This is where I get to see myself teaching, which is always a bit weird. We'll get to see Michael's dog again. All right. Okay. So we ended up here, types of transposons. Okay, you can stop. Don't need to hear that anymore. Right. So that's right. Yeah, we didn't have time to start on retro transposons because those are kind of funky and fresh. Okay, so basically we talked about different types, right? And so most DNA transposons are mostly prokaryotic or found in prokaryotes, much less common in eukaryotes. They can be both replicative, in which they copy themselves, or what they call cut and paste, because they literally cut it from where they're at, or cut themselves, really, right? Because they're self-mobile elements, cut themselves from somewhere and then reinsert themselves somewhere else. Remember, leaving behind that footprint. It's called the excision footprint, right? So that's the doubled up of that in target site. Remember the flanking direct repeats, which are created by the insertion of the transposon. Hang on, this bit's here, right? So those flanking direct repeats are left behind when a transposon is excised. That's why it's called the excision footprint. So if you can kind of like put your hand in front of the screen, like kind of like that, basically if you take the transposon out, you have to close one eye as well. Take the transposon out, that would be the DNA that's left behind, right? It would be just those flanking direct repeats. And obviously that's a doubling up, right? Because there was originally only one of those sequences. That's why even when transposons leave and go somewhere else, they still are mutagenic, right? Because that extra repeat on either side, whichever one, it doesn't really matter which it is, right? Is DNA that was not originally there in the host genome before that transposon transposed into that site. Anyway, so that's what cut and paste transposons are. There are also like really funky transposons called retrotransposons, right? And these are much more common in eukaryotes. It's also one of the reasons why we have just this huge amount of these things cluttering up our genome. And nearly half of our genome consists of transposons. And one of the reasons why is because these copy themselves, right? They don't jump around and move around like cut and paste transposons do. They stay put and make a copy of themselves that then gets inserted somewhere else. So you can see, obviously, once that happens, then both of those sites can now copy themselves and insert again, right? So now you have this potentially exponential increase in the amount of transposon DNA in the host genome. And so retro, and this is actually another, I kind of talk, not sure if it was you lot or the other lot, about my kind of fondness for dogma breaking, right? Scientific discoveries which turn, our understanding on their head, essentially. They're kind of, in my mind, the most interesting discoveries of all. And so this is one of them. All right, so you've all heard of the central dogma, right? We're going to start talking about it probably when we finish this lot. And so the central dogma actually is not what you think it is. This is kind of like a pet peeve of mine. I have all my peeves next to me. This is one that's a pet. I don't know if you know that phrase in the US, but anyway. So pet peeve of mine is that the central dogma does not refer to gene expression. That's the sequence hypothesis. The central dogma actually refers to the theory or the dogma, right? Unfortunately chosen word, that you cannot go from protein to mRNA or from mRNA or RNA to DNA. That's what the central dogma actually means. When Watson and Crick penned that back in the mid-50s, that's actually the meaning of the central dogma, right? And so they actually quite frankly said, well, we may be wrong, right? Because we don't know all this stuff. We've only just discovered the structure of DNA, right? So don't expect us to know everything else. But we're positing that it only goes this way, the sequence hypothesis, DNA to RNA to protein, not this way, backwards, right? That's the central dogma. And so retro stuff, and we're not talking about like bad hairdos and like genes that come up to your nipples and stuff like that. We're talking about retro in terms of molecular biology. That means going from RNA to DNA. And I'm going to put molecular biology here just in case you get confused. Means going from RNA to DNA, and that uses an enzyme called reverse transcriptase. Now I could even tell you what, like commercial one, Invitrogen Superscript 2. Haha, I've used it so much that I even know what it's called off the top of my head, right? And so this is actually something that I've used a ton of in my career and my research, right? Because if you want to study RNA, RNA is literally like a snowflake. You look at it and it disappears, right? And so to work with it, you have to turn it into DNA. And to do that, you use an enzyme called reverse transcriptase, right? And so this is actually something that broke the central dogma, right? Because this proved that you can actually go from RNA back to DNA, right? We still can't go from protein back to RNA, and you'll see why when we get to translation. There's a good reason for that. But you can go from RNA back to DNA. And so, here's a question for the class, because I've been busy running around all morning. Anybody think of a infectious agent and actually has retro in its name that uses this enzyme? Part of its replicative cycle is to get turned into DNA. Give you a hint, it's called a retrovirus. Big issue from like the 80s, 90s onwards, still a major problem in Africa and amongst drug users, for example. Less so now though, thankfully, because there's a whole bunch of.
Was it HIV?
Yeah, HIV, which causes AIDS, which is the syndrome of all of the consequence of not having any T4 cells, basically CD4 plus cells, or yeah, CD4 plus cells. So yeah, HIV is a retrovirus, as is varicella, which causes shingles. So you might have an old relative somewhere that has shingles. My grandmother did. But anyway, they work by their RNA viruses, right? And so they turn their RNA into DNA. And in the case of HIV and varicella, they actually integrate that DNA into your genome. So that's why it's very, very difficult to truly fully cure someone of HIV, because that virus can go dormant actually in your genome, right? So it's not even present as a viral particle in your body. And then it can just reemerge later and continue the replicative cycle. So anyway, that's a little bit of a tangent, but it's always a fun one to talk about for a bunch of different reasons. And so these transposons, these retrotransposons, they actually encode two enzymes. They're actually bigger and more complex than the simple DNA transposons. So retro encode transposase, right? Because I need to use that to do the transposition, and reverse transcriptase, because they need that to make basically their RNA kind of form, which is transcribed back into DNA. And fun fact, reverse transcriptase is one of the key targets of anti-HIV drugs. And the reason for that is that we do not have a reverse transcriptase. Humans do not produce one. So it's the perfect drug because A, it's super important to HIV's kind of, life cycle, I guess, what you could call it. And also we don't have one. So you can bomb the crap out of it and it's not going to harm the host. So anyway, fun facts. So anyway, that's a bit of a digression. No, not transgression. That'd be if I did something wrong. Digression. There you go. And retrotransposons have a really weird way of moving around. So basically they have the same structure, right? We have flanking direct repeats. We have these terminal inverted repeats, right? Which are required because those are recognized by the transposase. And we have the body of the transposon, which has those coding regions for the transposase and the reverse transcriptase. However, what happens first is that gets transcribed, right? So it gets turned into RNA by the host transcription machinery, as if it were a gene being transcribed. And once we have that RNA, now it's going to be reverse transcribed, right? So it's going to go back into DNA to create a piece of double-stranded DNA. And that double-stranded DNA now essentially looks like a transposable element, as the terminal inverted repeats recognized by transposase. It has the coding regions in the middle. And so now it does the same process as we've talked about before. Transposase makes a staggered cut in the target sequence, right? Whatever it recognizes. Inserts the new copy of the transposon, right? So the old one stays put. It doesn't move because it's made a copy of itself. And in the process of that transposition, now we create more flanking direct repeats. So now you can see how you can get from, I love this thing that tells me when I'm getting scammed. I think it's fabulous. No longer do I have to answer phone calls asking me to extend my car's warranty. So now you can see why we can expand the amount of DNA that consists of these transposons, because every time one is transposed or copied, now you have two, each of which contains the ability to make more copies of itself. and so on and so forth. So that's part of the reason why different organisms have different genome sizes, is because some of them have more active and more of these types of transposons, particularly in eukaryotes. And so if they've, had enough time to do all their crazy stuff, then you have a bigger genome, right? Even though you have the same number of genes or genetic complexity in terms of transcription alike as that other organism. So as we've talked about a little bit before, transposable elements or transposons are always mutagenic, right? They're mutagenic when they insert or transpose, because you've got this big chunk of DNA that's now in the middle of wherever it's gone, right? And frequently transposons insert in or near protein coding regions. And even when if that transposon leaves or is inactivated by mutation, right? So no longer kind of functions as a transposon, it still is new DNA sequence in that target site. And anytime you have new DNA sequence, it's a mutation, right? Whether that's beneficial, neutral, or deleterious, it doesn't matter. It's still a mutation. And so over half of all spontaneous mutations in Drosophila are due to transposons. And actually, Drosophila has been the main organism in which transposons have been studied, because it has so many of them, right? And they are very mobile as well. Piggyback, the one that I worked on, was originally discovered in Drosophila, for example. And just to finish up, so transposons can create kind of what one would call small scale mutations, right? They might insert into a protein coding region, which equals no protein. They might insert into a promoter region, which may change the control of the transcription of that gene in who knows how many different ways, potentially good ways, potentially bad ways. Or it just may insert into somewhere where it does absolutely nothing. But they can also create really large scale mutation. And so I'm not talking about change of genetic sequence, I'm talking about change of chromosome structure, right? And so it's thought that transposons are the cause, or yeah, I guess the cause of some chromosome duplication and merging and splitting events. So if you look at the chromosomes of related species, like us and chimpanzees, for example, you can actually see where two of the chromosomes that we have, that presumably our last common ancestor had as well with chimpanzees, have actually merged and formed one much larger chromosome in chimpanzees. And I think one of the reasons for that is that these transposons have the identical sequence. And so if you copy one of those transposons into another location in the same chromosome or into a different chromosome, those can actually serve as a site of homologous recombination. And so now, instead of having, chromosome 1 and chromosome 1 line up like so, now chromosome 1 might line up with itself, in which case weird shit happens. Like you can actually get part of the chromosome flipped over in order. Or chromosome 1 might line up with chromosome, 20, because they share the same sequence now because of the transposon. And now when you have crossover, now you get something really weird, right? And there are all kinds of weird things that have happened in our evolutionary history that have been caused by those sorts of events, like different genes getting stuck together and creating a new protein, for example. So like gene fusion and stuff like that. It's very, very strange. Anyway, so that's it for transposons. You can see why, I spent a bunch of time talking about them, because they are very weird and they're very I think they're really important in terms of our evolutionary history as well, right? Both how, our genomes have evolved and how we've evolved as well, right? In various different ways. So they're really important things to talk about, even if maybe you'll never come across them again in your own personal scientific lives again. Who knows, maybe you will. All right, so that's that from that. Now I've got to open up. Doop-a-doop-a-doo. Shum-de-dum-de-dum. The next one, because I haven't had time to do that because I was on the phone with a Spectrum tech. Doop-a-doop-a-doo. Where are we? Ha ha ha. The meat and potatoes, as my dad would say, of the course. All right, so this is the big one. Actually, it's not the big one. That's control of eukaryotic gene expression. That one's a monster. Give you a little, how do I get this smaller? There we go. This one only has 28 slides. Needless to say, we're not going to get through this today. All right, so essentially this is going to be the subject as a whole, right? That's going to take us to the next exam, exam 3. And so I will tell you now. Oh, I'm sorry. I've had far too exciting of a day so far. I need to go lie down. There will be a big amount of details to learn. So this is one of those things that you cannot just wing. You have to put in the footwork, legwork, whatever you want to call it, to really get this stuff down and understand it. And so one thing to be very, very aware of, and actually I think generally speaking, people have been doing pretty well so far in terms of exams and stuff. Actually, I looked at the exam scores, I was like, damn, do I need to make the exam harder? People are doing too well. Anyway, sorry, that's a professor joke. No students ever laugh at that because they're like, what? No. But this one's going to be a lot of details. So don't, how can I put it? Essentially, you don't want to fall into the trap of nodding along to what I say because it makes sense while I tell it to you. And then get into the exam and go, whoa, I'd actually forgot that key part. I don't understand that, right? Or I kind of understood it when he was telling me, but I don't really understand it now on my own, right? Because I'm not going to be there to help you in the exam, right? So that's a key point. It's actually very, very common in my students, and I've taught this course a lot. I actually enjoyed teaching this course, so it's not a major deal, but I have taught this course a lot. And quite often what happens, particularly in exams 3 and 4, which are very, very detail heavy, is students have this false sense of security that they know stuff. And then they get to the exam and they realize they don't actually understand it as well as they thought they did. Right. And that sucks, basically, for everybody, right? Because it means that you're going to get a bad exam result. And I don't want that to happen. So make sure as you go through this stuff, particularly, gene expression and control of gene expression, because there's a lot of there's actually some fairly complex topics in here, right? Make sure you understand this on your own. Get people to quiz you, quiz other people, right? Try and write out stuff on your own without access to your notes or any kind of online resource, right? Make sure you figure out what you don't know before you get into the exam. Right? Otherwise, you will be unhappy, right? And so the exam is not designed to, trick you or piss you off or catch you out. But there are a lot of really, really important details in these 2 sections, without which these bigger concepts don't make a whole bunch of sense, right? And so I'm telling you this, because quite often in my SRIs, I have students complain about exams being too hard or on points that they thought were buried in PowerPoints or mentioned only a few times in the Zoom sessions, right? And again, I'm not trying to catch you out. It's just all of this stuff is important. Otherwise, I wouldn't be telling you it, right? Other than my, random asides and, children's film references and stuff like that, right? I actually got one in for WALL-E in lab the other day. Contaminant. Don't know if anyone recognizes that. That made me laugh. Anyway, so what this is all about is essentially the process of going from the information that we have in DNA, the information that's inherited by all what I call living organisms, viruses accepted, and turning that information into something useful. Typically, that something useful is protein, but not always. It can also be RNAs, things that are called non-coding RNAs, such as ribosomal RNA or tRNA, and there's like this whole family of them. Pee-wee interactin RNA, small interfering RNA. There's actually more, but I can't remember them all, to be honest. Small nuclear RNAs, those are always fun. Anyway, so there's a bunch of stuff produced, and it's all produced in a very particular way. It has to go from DNA, that DNA has to be transcribed into RNA. If it's non-coding RNA, that stops there, right? If it's coding RNA, that then has to get potentially processed if it's a eukaryote. If it's just a prokaryote, it's got to get translated, right? If it's a eukaryote, it's got to get processed, then translated, right? And potentially, even after translation, you're going to have other processes which turn that newly made protein into a functional protein, because that's what we care about. Functional protein. Now, it might be functional in the sense that it's in the right place, right? Because if you have a protein in the wrong place, it can't do its job. It's not functional. It could be a protein which has to be processed, kind of like chopped up, like sushi, to be able to give you the functional protein. Or any combination of that stuff. Might need to have stuff added to it, stuff taken away from it, all kinds of things, right? There are lots of that whole aspect of gene expression is a huge and complicated one. Actually, all of this is huge and complicated, right? It's a phenomenally complex process, right? Because it's a phenomenally important one, right? This is the basis of all of your biology, right? If you've had, I'm actually thinking about coffee right now. But if you had a nice breakfast, I did. I like mini wheats and banana and yogurt and stuff. It was little did I know how my day would turn out while I was eating my breakfast. But anyway, if you've had a nice breakfast, right, you digest that food, blood sugar goes up because all of the glucose and the like gets absorbed out of your gut into your bloodstream. And then your pancreas goes, whoa, hang on, this ain't good, right? Because high blood sugar is not a good thing, generally speaking, right? And so your beta cells and your pancreas release insulin. Insulin then kind of, wends its way around your body and tells your muscle cells and your liver cells to take glucose up out of your bloodstream. And so that brings the blood sugar levels down. And basically you want to kind of keep it within a couple of bounds, right? It's called homeostasis. We do an awful lot of it. Temperature, for example, right? Et cetera, et cetera. So that protein, insulin, is a small fragment of the protein produced during gene expression. It's cut up twice. It's packaged into little vesicles, which are like little containers, essentially, cellular containers. And those are stored in your beta cells until your beta cells get the signal that your blood sugar is too high. Then your beta cells release them. Literally, they merge, like they kind of go stunk, and they merge with your beta cell membranes, release the insulin. Those insulin or small bits of insulin protein travel around your body, bind to insulin receptors on your muscle and liver cells, and activate them, right? So even that relatively simple part of your daily life, right? Having breakfast, digesting your food, your blood sugar level goes up, relies very, very tightly on this whole process of gene expression, from turning on your insulin gene, producing insulin mRNA. That insulin mRNA gets processed because we're eukaryotes. That insulin mRNA gets translated into protein. That insulin protein, which is actually called pre-pro-insulin, gets sliced and diced, and then eventually it gets released into your bloodstream, and now it is functional, right? So you can see there's a long series of steps between the DNA, the information, and the protein that does stuff. That's also why your beta cells stockpile insulin, because there's no way you can do all of that sufficiently quickly to bring down your blood sugar level back to safe levels after having, like, you know, a quart of ice cream or something, right? It's also one reason, not the only reason, because diabetes is super complex as a disease, but it's also one reason why you get diabetes if you have an elevated blood sugar level for a long time. Essentially, you wear out your beta cells and they die because you're asking them to do this whole process, devote so much of their energy into making insulin because your blood sugar is always high, right? You got constantly releasing insulin that your beta cells just go, sorry, and they cark it and they die. So you no longer produce insulin, and then you start having the issues of elevated blood sugar. That's one of the reasons. It's not the only one, but it's one of them. Anyway, so there's a lot to this, right? It's why we're going to devote a whole bunch of lectures to it, right? Actually, I can't even remember how many, a bunch of them anyway. So I'm going to go through this in steps. We're going to outline kind of like the general principles of this. And then we're going to look at how it works in prokaryotes, because prokaryotes have their own wrinkles. And we're going to look at how it works in eukaryotes, which have their own peculiar wrinkles. And as you can see, this is gene expression, a fairly simplified version of gene expression in eukaryotes, because we have a nuclear membrane, i.e. a nucleus, right? Anyway, so here's some definition. Basically, it's the way in which you produce the things that determine your phenotype. At the end of the day. And that could be phenotype in terms of what you look like, could be phenotype in terms of how you respond to something, could be phenotype in terms of what things look like when they don't work right. You know, all of that ties into gene expression. That's why really at the end of the day, it's the underlying basis of all biology. Right, because whether you study giant pandas, much as I hate them, you're still studying the consequence of the gene expression in those useless, giant, fluffy animals, right? There's still gene expression going on. And so here are some of those RNAs that I mentioned earlier. We're not going to look at all of them, but we actually will look at some of these. And so these and proteins, right? We have, I don't know, around 20,000 known genes, protein coding genes. We have an unknown number of non-coding genes. And we don't know how many we don't know we have, right? Because they're very hard to find. So basically, and this is kind of part of the overview, some of you may already know this pretty well, but it's worth going through again nonetheless, is you have a series of steps. These don't always happen in the same place. They don't always happen at the same time, right? So there are many variations in terms of time and space on these steps. Prokaryotes generally being simpler, eukaryotes being much more complex. So the first step is transcription. That's the creation of RNA from a DNA template. As you probably guessed it, you're creating an RNA polymer. So the enzyme that does that is RNA polymerase. This whole jazz here is the actual process of transcription is not super complex. The control of transcription certainly is, right? So control of whether or not a gene is transcribed. Because essentially, if a gene is transcribed, it's being turned on. And so you only want to do that if you need the product of that gene, right? So that control of that is very important. So in eukaryotes, that RNA has to be processed for a bunch of different reasons, which we'll get into in a bit, kind of tied into gene structure as well as other aspects. And then that processed RNA molecule, or unprocessed in prokaryotes, is used as a template itself to make protein. And so the sequence of the transcript contains the information for which amino acids are going to be used and in what order. And those amino acids, when joined together, are called a polypeptide, right? Because essentially when amino acids get joined together, they become peptides rather than amino acids. And so when you have a long chain of them, that's a polypeptide. Now, this polypeptide is not necessarily functional. There are many other steps that may follow on from that. Folding, processing, so on and so forth. Right. But this is the general kind of somewhat simplified overview of gene expression. Now, gene expression is also directional. So remember, we banged on about quite a lot, actually, about how DNA has direction, right? The whole 5' to 3' deal. And it can only be made in that direction, right? Because you can only add new nucleotides onto the 3' end of a nucleic acid chain, essentially. So that directionality kind of goes through all of the steps. So DNA is directional. So the mRNA, which is synthesized using the DNA, is directional too. And is also specific to the direction of the DNA that it was transcribed from. So this particular fragment down here, this mRNA, this has the same sequence, but in mRNA, of this strand up here, the top strand of this double helix. Because this top strand is going from 5' to 3'. This bottom strand is going left to right, 5' to 3'. However, the strand that's being copied, and we'll kind of come back to this again as well, the strand that's being copied is not the one that's being used to make the mRNA, right? Because it can't be. You can't make mRNA exactly the same as the coding strand using the coding strand. you have to use the template strand, which is the other one. So the template strand is the strand of DNA going in the opposite direction to the one which is being copied. Again, we'll come back to that because it's a little bit of a, once you get it, but it's a little bit of a tricky concept to get just by describing it. Anyway, so that directionality of mRNA continues on into the directionality of the polypeptide. All right, so proteins have a beginning and an end. Their beginning is the amino terminal or N terminal, and I'll write this down because it's important. And it's called the amino terminal because of the N in amino, also because amino is a nitrogen and a couple of hydrogens. So the N terminal is the beginning, the end is the carboxy terminal or C terminal. And the reason for that is because there's a carboxyl group, right, at the end of the last peptide in that polypeptide chain. So that directionality flows all the way through this process. It also means that you can actually read DNA in both directions. This is actually something that's a little bit kind of trippy to get your head around, right? We have two strands of DNA. Both strands can be the coding strand, right? So we have one DNA strand at the top, one at the bottom. This one can produce transcripts going that way. This one can use transcripts going that way. So you can have genes on both strands of DNA. And so when we talk about a gene, what we're really talking about is the information needed to make the product that that gene encodes, right? And so the information for this particular transcript, right, is encoded in this strand here. The information for this transcript is encoded in this strand here, right? So you can have genes going in different directions, right? Because all you care about is the coding strand, right? The sequence of the non-coding strand, the template strand, is there purely to allow you to make the mRNA. So actually, even recently, and this was a kind of a new thing for me, I found in one of the genetic backgrounds I'm interested in, a couple of genes are highly expressed, right, compared to normal, and they share a region. So they're actually like that. So there's a region in between them, I think it's about 1,000 base pairs, give or take, which is common to both of them. And that's super interesting because that means that those two genes may be controlled by the same protein to be turned on or turned off. So when I saw that, I was like, ooh, that's kind of weird. And suppose it's fairly common. Anyway. So there's some major differences between prokaryotic genes and eukaryotic genes. So prokaryotes have a pretty simple gene structure. They have a beginning, they have a middle, and they have an end, right? So this is TRPA, this is a tryptophan gene, or a gene involved in the synthesis of tryptophan, right? So it has something to control it, has a protein coding region, which is what is shown here, and it has something to stop it, essentially, stop transcription. And so the amino acid sequence, right, the polypeptide maps exactly to the DNA. So this, what we showed you up here, is pretty much for a prokaryote, for a bacteria. So the sequence of DNA gets copied into the sequence of mRNA. The mRNA is used to make polypeptide. So they are absolutely kind of collinear. So if you screw up this bit of DNA here, you'll screw up this bit of mRNA here, you'll screw up the protein here. That's essentially what we're talking about. Right, and this is basically shows that, if you have a mutation in DNA here, it maps directly to a change in the amino acid in the polypeptide sequence. Now, eukaryotes are very different. They are not collinear at all. So eukaryote genes have this kind of fairly funky structure, right? They have a whole bunch of the gene which is not actually transcribed or translated. And so we have the start, just as we do with prokaryotes here, right? We start here, but now we have what's called UTR. So UTR is translated region. If I'd spell it properly, that's what it should look like. So this is part of the transcript which is not turned into protein or used to make protein. Well, does not code protein. There you go. You got to be so precise talking about this stuff. And now we have two new things. We have exons and introns. So exons are protein coding. Introns, things in between exons. And those are non-coding and are removed before translation. So every eukaryotic gene has at least one intron. You never find a eukaryotic gene without any introns. Karyotic. And these introns can be huge. So typically, by and large, I don't know, a prokaryotic gene on average maybe say 3,000 base pairs, say 3 kilobases. The coding region of a eukaryotic gene is on average about the same. But the actual space occupied by a eukaryotic gene can be in the tens of thousands. They can be huge, right? And the region for that hugeness are the introns. And we'll get into some of the reasons why we have introns later on. But I think that is probably enough for today. We're at time. So, pretty wild. Sorry about the whole craziness at the beginning. I guess it's just one of those things in an internet world, right? We will be able to catch up over the course of the next few lectures, so don't worry about that. I would, as always, nothing different here. Read ahead. Reason being is we're going to use a lot of different language and terms like coding strand, template strand, introns, exons, UTRs, promoters, so on and so forth, which may be new to you, but they're essential to understanding what I'm talking about. So even if you don't understand what you're looking at completely, at least you would have seen it before we talk about it. So you'll be a little bit kind of clued in, I guess, to what we're going to be talking about. Because this, and I'm not going to like sugarcoat the whatever, this is probably the hardest section of genetics. This one and the next one. Actually, probably control of gene expression is even harder. So it's really important to kind of get your focus straight. All right. Thanks very much, everybody. It's been a fun day. Gonna go have myself a cup of coffee now and just kind of relax. I'll get this stuff up in a bit and have a lovely rest of your day. I'll see you next week.